Oh boy. So I just posted active notes for Chapter 15, Part 1. It's going to be a short lecture because I attached like some follow-up questions at the end, which I will grade this time, and we'll just see how that goes. So you may want to pull up the active notes.
Are you going to grade the notes part?
Not the good notes. I'm just going to grade the questions that I posted at the end.
Why doesn't it just like go through? Bruce tells us how to type out types. So now we know how to type out types. I don't know how to do that.
So you can't roast it.
Yeah, I know. I still will.
I've also inserted questions like in between slides that I'm going to ask you, so just get a little bit more interaction. I got some feedback from Herman Norcross, so we're going to see how this goes.
Wait. We'll send a copy and then I think we can open it like that. Oh, I just broke.
It's being really slow.
Well, well, I suppose.
Is this chapter, what chapter is it? No, I don't think this is very long. Gene expression.
We open it on
Is there, do you say you want us to open it?
If you want, or you can, I guess, just watch the lecture later, but the questions are at the end, so you're going to have to open up the document anyways.
No, I mean like to write.
Is it only 22 slides?
Yep, yeah, it's shorter. Shorter than mine because I don't think I put the questions between slides. Okay, so chapter 15 is gene expression and how genes get expressed. So chapter 14 was talking about the DNA structure. So we're just going to build off of that and eventually get to the DNA dogma, how information flows in a cell. Oh, why did a slide do that? Skipped, like, again, 4 slides. So we know that DNA is the genetic material, and we know the structure of it due to Watson and Crick. So some questions to ponder is, how does this information actually portray to the traits that are found in organisms? Really, what does a gene actually say? What does it actually code for? And how is this message translated by the cells to that specific trait? Maybe for hair color, eye color, different blood types. So remember Gregor Mendel in his pea plants, he was looking at height in one case, one of the 7 traits, tall versus short, in this case dwarf. And so a variation in a single gene can account for these 2 differences, tall, dwarf. The pea plants that were dwarf, people started to analyze that and say, well, why is it short? And they found out that these dwarf plants lacked the hormone gibberellin, which helps with elongation of cells. So then what they said is, okay, if the dwarf pea plants don't have gibberellin and we give them gibberellin, what's going to happen? So they took the dwarf pea plants, treated it with gibberellin, and these pea plants were no longer dwarf, they were tall. So kind of the first piece of evidence that shows that genes code for chemicals, maybe a protein, an enzyme, or in this case, a hormone. So then another question is said is, okay, so why do these dwarf plants lack gibberellin? How come they cannot make their own? They're missing a key protein, in this case, an enzyme that helps make gibberellin. And so the main point is that DNA, yes, is the genetic code. It leads to specific traits, but what causes those traits are usually proteins or enzymes that help develop that phenotype. So proteins are the link between our genotype and our phenotype. The genotype are the alleles, dominant, recessive. The phenotype is what you actually look like, physical traits that you can see and in between are the proteins that make it happen. So today I want to talk a little bit about the history behind making those connections between genes and enzymes/proteins. And then I'm going to talk about the flow of information from DNA to RNA to proteins, and then there are some exceptions to some rules. Talk about how this genetic code was cracked, and then give you a very broad overview of gene expression, which we'll dive into next week. So our story begins with Archibald Girard, who was looking at disorders that were passed down to each generation. So basically he was studying pedigrees and families, and he noticed that some diseases were very prevalent in particular families, and this is the early 1900s. And the diseases that were being studied were simple recessive alleles, which means if you had the disease, you were, little r, or whatever, but you were homozygous recessive. And so, luckily for him, the diseases he was studying, they followed the Mendelian traits of either or either this or that. One disease that Archie studied was found in urine, in this case, alkaptonuria. And basically what happens is these patients, their urine isn't that nice yellow color. They pee black urine, which can be pretty scary. Really? I mean, could you imagine? So he noticed that these patients that had black urine had large amounts of a chemical called homogenistic acid, or alkapton. And so if you have a buildup of this in your urine, you have alkaptonuria, which means that your urine will be black. So what's happening is that this chemical undergoes a chemical reaction when it's exposed to air, turning that urine black. If you have normal urine with the yellow, this chemical gets broken down into simpler substances and it turns out to be yellow. So he's asking himself, okay, why? Why does this happen? Well, those alkaptonuria people lacked that enzyme that catalyzed the reaction to break down this homogenistic acid into something simpler. So they lacked the enzyme that could break this down into simpler substances. So he's actually one of the first people to make the connection between genes and chemical reactions. So think back to Chapter 6 with metabolism, chemical reactions that take place inside the cells. A lot of times, chemical reactions are a metabolic pathway where you have a series of enzymes kind of all lined up in an assembly line where you take one substrate, latches onto the enzyme, it gets modified, bonds are rearranged or broken, or things get added to it. You get a new substrate. This substrate only reacts with this enzyme, again, rearranged, whatever, to a new substrate, and so on and so forth. Well, in this case with alkaptonuria, we have this chemical, p-hydroxyphenylpyruvate, that interacts with an enzyme to turn into homogenistic acid. But then these people lack this enzyme that breaks it down into something even simpler. So they didn't have this enzyme, thus this chemical builds up. So I did a little cartoon diagram. They don't have this enzyme, so that homogenistic acid builds up in the urine, and then when they micturate, pee, gets exposed to air, turns black.
Can I get some black?
You should. Some other scientists that were, kind of following in Archie's footsteps, Beadle and Tatum, 1941, so several decades later. Everyone's trying to figure out what genes code for. So they said, okay, what if we just mutated genes and just, see what happens to the organism? We know what they should look like. What if we mutate certain parts? We'll know what it codes for. So they used bread mold, a fungus that can be grown on a minimal medium in a petri dish. And so they took the wild type bread mold that's normal, okay? They exposed it to x-rays because they know that x-rays cause mutations. They grew them in various different mediums, and they looked at whether or not they grew or not. So no growth or growth. And so this table kind of simplifies their experiment. Here we have 3 different mutants, and they just called it ARG1, ARG2, ARG3. And the reason for that is ARG is the abbreviation for arginine, which is an amino acid. So with ARG1, they noticed that if you didn't give it any supplements, any nutrients, it didn't grow. But if you gave it O, it grew. If you gave it C, again, so these are just various nutrients, it grew. And ARG, it grew. With ARG2, no supplements, didn't grow. If you gave it O, it didn't grow. So like, whoa, what's going on there? But then C and ARG, it grew. With ARG3, it only grew when you gave it ARG. Now you notice this is like a nice little stepwise fashion. So then what they did is they could figure out the chemical pathway of these enzymes. Some of these enzymes were unaffected, some were. So here's this little flow chart here. You have a precursor molecule that gets modified by an enzyme that turns it into O, and then O reacts with this enzyme, turns it into C, and then C reacts with this enzyme and turns it into ARG. So with the mutant 1, if it can grow, given an O or a C, it's got to have enzymes 2 and 3, but maybe it lacks enzyme 1. With ARG2, if you gave it C, it grows, but not with O. So therefore, it has enzyme 3, because it grew in this medium, but not enzyme 2, because if you just gave it O, it didn't have that enzyme, it turned it into C.
So the difference between all those is just the enzymes?
Yeah, the defect in the enzymes. Yep. So then with ARG3, it could grow if you gave it ARG. But if you just gave it O or C, it didn't grow because it had mutations in enzyme 2 and 3. So they kind of like work their way backwards, cause the mutations, work your way backwards, figure out the chemical pathway, and then, oh, we mutated these genes that code for that enzyme. So with Beadle and Tatum, they could call it a one gene, one enzyme hypothesis, okay, but that got changed to a polypeptide. So at first, Beadle and Tatum said, okay, genes code for enzymes because they were looking at chemical pathways. But we know that not all proteins are enzymes. Like hair, skin, they're made of keratin. That's a protein, not an enzyme. Hemoglobin, which is found in red blood cells, that's a protein. It's not an enzyme. So you can't say one gene codes for one enzyme. Sometimes it does, but not in all cases like this. So then they had to modify it and say, okay, one gene codes for a protein, but sometimes proteins are made from many polypeptides. So you have to think back to like chapter 2 with chemistry, the structure of proteins, where you have the amino acid sequence, and you have that hydrogen bond in, and you have tertiary and quaternary structures. Well, to get to that quaternary structures, there are many polypeptides that it's interacting with. So you can't say one gene, one protein, but you can call it one gene to one polypeptide. Now, I know in my lecture and in future reference, I'm probably going to say a gene codes for a protein. But just know that really it's a polypeptide that a gene codes for. Okay? So I'm just going to use these interchangeably, probably this one more than this one. Okay, so a question for you, what do genes code for?
Polypeptide.
Polypeptides.
Wait, so do we not have that slide?
Right.
Oh.
They're just questions.
So we don't have to fill those out anywhere.
Nope. Okay, moving on to how information flows. The central DNA dogma. DNA gets converted into RNA or is translated to RNA, and then RNA is used to make proteins. But how do we know this? Okay, so we have to look at the structure of our cells. We know DNA is held in the nucleus. And then scientists knew that ribosomes were found in the cytoplasm. But if we know DNA codes for, in this case, polypeptides, how do the ribosomes get that information? So the hypothesis is that there's this short-lived molecule that goes from the RNA to a ribosome. So in order to seal the deal and actually prove this hypothesis correct, what they did is they took a DNA strand that was just made up of a bunch of thymines, put it in a test tube, inserted RNA polymerase, which is very similar to DNA polymerase. DNA polymerase makes DNA in the process of replication. RNA polymerase makes mRNA, or RNA. And then some ribonucleotides. What are the 4 ribonucleotides in RNA?
U, A, C, and G.
Yep. U, A, C, and G. Okay. All unicorns can giggle, right?
When's the T?
T is DNA.
Um,
And then they said, okay, so the strand is TTTT. Let's just see if an RNA strand, made up of a bunch of A's, is made, and it was. So they know that RNA is this intermediate molecule that transfers that information from DNA to the ribosomes to make proteins. So information flows in one direction, but in eukaryotes, sometimes it can go backwards. Okay. RNA can be converted back to DNA through a process called reverse transcriptase. Viruses usually do this. So if you ever see the term retrovirus, maybe they're talking about a certain virus and they say it's a retrovirus, that means that it takes RNA and it works its way backwards to get DNA. So it uses RNA as a template to make DNA. So how does genetic information flow in a cell? I'm just going to draw a diagram here. So what do you start with?
No. What's that?
5 and 3.
the DNA.
DNA.
And then RNA.
Well, that's what you're saying.
proteins.
Okay.
Yeah.
I'm going to ask you a question you may not know. What is the process of going from DNA to RNA? Does anyone remember from 10th grade biology?
Transcription.
Yeah.
Good deal.
What was the one from RNA to DNA? Like reverse transcriptase?
Right. Thank you. That was going to be my next question.
Oh.
So that's why they call it reverse transcriptase. And then the process of RNA to proteins.
What's it start with?
T.
Translation.
Translation.
Thought about guessing that just
Yeah, that was kind of sad.
Okay, now chapter 14 talked about this process where you have DNA and it makes more DNA. It's another word for copies,
Replication.
replication. So this is what chapter 14 and 15 are in a nutshell. So chapter 14 talked about DNA replication. Okay, chapter 15 we'll talk about transcription and translation.
For some small fleeting moment, I thought that maybe we were done.
Almost.
And I looked at the clock and I was like,
I mean, it is a shorter lecture. I mean, you'll have time to work on those questions and ask questions. All right, so moving on to the genetic code. This is the last section I'll cover today. The code is read as a triplet. And so now you're probably wondering, well, how do we know that? Right now, as far as we know, there are 20 known amino acids that are found in living things. And we know that DNA has 4 bases, which are,
ATGC.
yep, A, T, C, G, yep. Okay, so then they said, okay, if the code is just one nucleotide for one amino acid, that means we can only have 4 amino acids, but there's 20, so that doesn't work. If the code is 2, there are 16 possible combinations, but we have 20 amino acids, so that doesn't work. So they put 4 to the 3, which gives you 64 possible options. So that fits the 20, okay, and then some. So the genetic code is actually redundant, which means that there could be multiple codes or triplets that code for the same amino acids. Okay, so if you ever come across the word redundant, that means for leucine, maybe there's 3 different codons that code for leucine. Next question is this triplet code spaced or unspaced? So I have this makeup sentence. Why did the red bat eat the fat rat? Okay, and so I grouped it into 3. So this is a triplet code, if you will. Pretend that these are nucleotides. So if I were to take away the T in the, okay, scientists started to look at the end result of the protein. Does the read-in frame just like read it like that? And even though it knows it's missing one, does it keep it the same and the only thing that's going to be different is this amino acid? Or does everything get shipped over and now instead of getting the the sentence, why did the red bat eat the fat rat? You get, why did her, so these two are okay, but then you get a new word, maybe it's going to be okay, and then all of a sudden, you get the 8 at hef at at. Okay, so when they
That's what it does. Is it messes everything up.
it messes everything up. So the read-in frame is unspaced. It just reads each 3. In this case, with this mutation, this is a deletion. And if the deletion is early in the sequence or early in the code, your protein is not going to be what it's supposed to be. Now, if the deletion was like at towards the end of a code,
Then you're gonna be fine.
it might be okay. Maybe it won't be as efficient. But that one little part's going to be a little bit messed up. So, it may not be the end of the world. We call the read-in frame a codon. Okay, so I know I used the word codon when I was describing this. So the codon is the read-in frame. It's just the 3 nucleotides that it reads at a time. So deletions early in the sequence, your protein's going to be really messed up. So we'll talk about mutations as well, and I think that's chapter 16. So in this diagram, if this is my DNA sequence, TAC, GAG, TAG, TCA, AAC, and T, the mRNA is complementary to it. So you get AUG, which codes for methionine, and then you get CUC. So remember, in mRNA, there's no T's. So it matches up complementary to it. This is the original amino acid sequence that you get. Now, this picture down here is what happens if you were to take away this nucleotide, like with a deletion mutation. Everything shifts over, and now take a look at the new amino acid sequence. It's completely different.
So, what does that mean? What happens when it gets messed up like that?
That means that protein's not going to work correctly. And if that protein is involved in a super important chemical reaction, like, people that are lactose intolerant, okay, that's not a serious one, but they can't metabolize lactose. Maybe that's caused by a deletion. I don't know. Okay, so with the code being a triplet, scientists like Nirenberg and a bunch of others wanted to figure out what each little codon coded for. Which amino acid does a codon code for? So they made synthetic RNA molecules in petri dishes, and they started with U. We have a string of U's. We add it to an in vitro system. We take a look at the proteins that are made. The result is they got a polyphenylalanine, which is an amino acid. And so then they just did all A's, and they did all G's, and they did all C's. And then they started to add, GUU or AUU, and they literally just went systematically with all 64 combinations. And they found out that 61 out of 64 code for an amino acid. 3 codons code for a stop, okay? And 1 codon only codes for methionine, which is an amino acid. So that's included in the 61. But they started to realize that all proteins, the first amino acid is AUG. It always starts with methionine. If I go back a slide, look at which amino acid it started with, methionine. It's always methionine. This this code is almost universal in all living organisms. These are the exceptions. In some single-celled eukaryotes, like parameciums, UAA and UAG code for glutamine instead of a stop codon. Yeast, CUG codes for serine instead of leucine. And then mitochondrial DNA is not quite the same, which is just more evidence for that endosymbiotic relationship where mitochondria at one time was probably a prokaryote. They got engulfed by a eukaryote, and now they live in mutual relationships. But with mitochondrial DNA, it's not the same. UGA is tryptophan, not stop. AUA is methionine, not isoleucine. And then they have some stop codons instead of arginine. So there are some exceptions, but it's almost universal, which is another reason why that maybe, all life started with, the similar origins, basically. So some questions. How many bases make up a codon? 3. How many stop codons are there? 3. All right, this last little bit is an overview of gene expression, which we'll dive more into detail next week. But it starts with transcription, which is going from DNA to mRNA. So next week we'll talk about how DNA is a double strand and how it unzips. And we have RNA polymerase that comes in and binds, but it only uses one side of the DNA. And the side that is used is called the template strand. And the side that is not used is called a coding strand. So don't get that mixed up, because I don't know why I would call it a coding strand. Because to me that'd be like, oh, that's the strand that's being used to make the code. No, it's the template.
Well, see, the scientists are smart in some ways, and other ways they're not.
Yeah, they just go out to make it as difficult as possible. Transcription does have 3 parts: initiation, elongation, and termination. Initiation means that RNA polymerase comes in, binds to promoters. We'll talk about what promoters are, and then how it makes mRNA. When it makes mRNA, this is actually the elongation phase. mRNA is made in a 5 to 3 direction, very similar to DNA. And then termination, eventually it will reach a stop codon on the DNA strand, which causes everything to kind of fall off, and the DNA rezips back up, and now you have an mRNA strand, and we'll talk about what happens to that mRNA strand. Translation is mRNA to the protein. Again, it has 3 parts: initiation, elongation, termination. Okay, initiation, that start codon. What's the start codon?
AUG.
AUG.
Gold.
How it latches onto a ribosome. We'll talk about it at an initiation complex. So it's way more complicated in eukaryotes than it is in prokaryotes. Then to elongation, where amino acids are carried by tRNAs. tRNAs stand for transfer RNAs. So we'll assemble an amino acid sequence chain, polypeptide if you will, and then eventually it will reach a stop codon, and again everything falls off, and then that protein or that chain of amino acids will fold into a protein. We're also going to talk a lot about RNA in the next couple chapters here. So mRNA is messenger RNA. Quick question for you. Why do you think they call it messenger RNA?
It's delivering the message.
It's delivering the message to the ribosomes of, hey, this is the amino acid sequence that you need to put together. rRNA stands for ribosomal RNA, and it's responsible for making the ribosome for translation. tRNA, T stands for transfer, and it just carries amino acids to the ribosomes. These 3 RNAs you may not be familiar with at all. So we'll talk about what they are. We've actually just kind of discovered these within the past 10, 15 years. snRNAs, SRP RNAs, and then microRNA or miRNA. So there are lots of forms of RNAs out there, and they all play a role in getting this message from the DNA across to make proteins. Okay, any questions so far? All right, so at the end of this lecture, there are 3 slides with some questions. Some are multiple choice. Okay, so all you got to do is write the letter, and you can just email me your answers. Some are short answer, as you can see here. This one, I give you an mRNA sequence, and you have to code it to the amino acid sequence. You're going to need your book for that, because there's a table of how amino acids get read. So use the table in your book.
Wait, don't you just use the opposite of what that is?
No, you're translating it into the amino acid sequence. So like, you're going to figure out what CCA, which amino acid. Is it leucine? Is it glutamine?
So it'd be like G, G, T, and then we find that sequence, or do we don't have to switch it first?
You look for CCA.
Yep, just look for CCA.
Oh, okay.
Yep. So you just need to find 3 amino acids.
It's pretty much normal.
Yes. Okay, and then the last slide is just 2 multiple choice questions.
Can we like write our answers on paper?
You can write it on paper and hand it into. Yep.
Yeah, I'm sorry.
So if you have questions, feel free to ask.
This is due on Monday.